Hello, good afternoon, ladies and gentlemen. I would like to thank the whole audience from the bottom of my heart for attending this presentation. And for those who have any concerns or questions on the back of their mind, I'll be more than glad to answer at the end of the session as well. So, before we get started, I would like to introduce myself. My name is Raed, and I'm a pharmacist holding the master's degree in clinical pharmacology. I moved to Canada recently, I believe in early 2021, at the middle of the pandemic. And unfortunately, since my credentials, they were not recognized, they have no merit as well. So I had to go through something called the Pharmacy Examining Board of Canada, which is an organization responsible for the, whenever someone is pursuing their pharmaceutical license, they have to go through PIPIC and perform like a three main big exams. And the good thing about it that I passed my first EE evaluation exam, MCQ qualifying part one, and I'm in the middle of preparing for my final OSCE exam on November. So wish me the best of luck here. So we do have a presentation today. It's called Human Genome and Personalized Medicines. So I actually didn't go through like an agenda here since I'm gonna try to minimize this as much as I can to stick into like 7 to 8 minutes. So let's get started here. Human genome and personalized medicine. So if someone asked, what is the human genome is all about? So human genome is the science or projects that study or mapping the DNA genes in general. So it tries to understand the replication of DNA, trying to have a better understanding of the nitrogenous bases that DNA have. We do have something called adenine, which shares between, DNA, RNA. We do have the thymine, which is related only for DNA. We have cytosine, guanine, which is shared between DNA, RNA, and finally we have uracil, which is only exists in RNA. So actually, healthcare professionals or scientists, they were into those genes, trying to have a better understanding of mutations, how truly takes place, and trying to link that with their medications or new invention treatments. So as we used to deal with long time ago with general medications, we used to target organs in general. We wouldn't have a better understanding of mutations and thus prevent any unexpected, proactively, disease in the future. So this is where we started to link between medication and human body DNA genes. So that actually improved, our treatment in terms of accuracy, diagnosis, try to diagnose the right, illness, have a better response, and disease proactively prevention as well. So let's get into the next slide here. We have a genome sequencing terminology. So what is genome sequencing terminology? It decodes an individual entire DNA. So as we discussed earlier, we do have those, what we call them the nitrogenous bases. And this is where we're gonna get through all gene individually, trying to have a better understanding of those mutations that takes place from time to another. So this is what causes disease, such as autoimmune disease. If we have like the lupus, if we have rheumatoid arthritis, it's like an autoimmune disease. This is where we have to have a better understanding of genes mutations to try to dedicate or try to find out the right medication. So this is how we can prevent like early prevention when we do have a proactively understand those genes and how they could really interact in the future and thus avoid any unexpected illness in the future. So actually this has improved our healthcare system really marvelously. So pharmacogenomics. Uh, Pharmacogenomics is trying to understand how those DNA genes is affected by the drug response. So the most specified drug we create, the most targeting genes and better understanding of those mutations that we can really, have here. So the healthcare professionals choose the right drug and dose for each person. So that thus reducing more side effects, improving treatment, and having a better outcome in terms of effectiveness. So, what is the scientific advancement? We have a huge breakthrough advance in terms of cancer treatment. We remember, like, back 25, 30 years ago, how we used to deal with cancers. We used to deal with organs, actually. We are treating organs. So we used to have, let's say that some patients, they have breast cancers, some of them colon cancers, lung cancer. So we used to target the organ. And this is why we used like generalized medications that causes a lot of emetogenesis as well, highly emetogenesis in terms of nausea and vomiting, such as doxorubicin, carmustine, cisplatin, which are like more than 90% emetogenic. And this is why we used to have those drugs such as, dexamethasone. We used to have also the lorazepam. We used to have a lot of anti-emetogenesis drugs. Like ondansetron, I believe, Zafran. There are a lot of protocols, medications to trying to minimize the emetogenesis. But right now, with the adventure that we have and studying the human genes, we could have a better outcome of those new medications, which targeting the genes themselves, such as bevacizumab, ranibizumab, which are considered monoclonal antibodies. Those are the new era of treatment. Even though the tyrosine kinase inhibitors, they are targeting a specified genes, specified cells. They are not used to kill the tumor cells and good cells as well, replication. No, they are only targeting the tumor cells, and this is thus how we really reduce the side effect, and at the same time we have a better effectiveness of drug. Okay, global benefits. Personalized medicine reduces the trial error prescriptions. So uh, let's admit that our healthcare system has really improved and it's been more organized. So in terms of cost effectiveness analysis, in terms of utilize effectiveness analysis, which actually we improved our quality of life on annual basis. We had this minimization effectiveness analysis. We have, we discussed about cost effectiveness, cost benefit analysis, minimized cost analysis, cost utility analysis. So thus, actually, we really improved our healthcare system according to those new medications treatments that we have. Okay, so now we have less drug-drug interaction. We have cost effectiveness, the highly cost effectiveness, that we are saving a lot of money. We are improving quality of life. So we have a lot of global benefits through this breakthrough studying the human DNA genetics. Global concerns, like everything has like a two sides cone here. Everything has its own pros and cons. So we have a lot of benefits, but we still have some negative things in terms of breaching of confidentiality. Because whenever we do have those studies, definitely it's not only it's going to be out of circle of care, right? Uh, there are a lot of insurers, a lot of employers who are responsible for those inventions. Actually, they will have our genetics, genomic codes, and they will be fully aware about some, patients who were really uh, let's say like those patients that they have been studied on. So it's a sort of discrimination. There are like some peers and genetic discriminations as well. There are breaching of confidentiality. So this is the only, from my perspective, this is the only global concern that we have mostly about it. But if we want to study the pros and the positive things that we have, it's way that there are a lot of things like benefits that we earned during or due to that genomic study. Okay. So that was everything. I don't want to I believe I reached about 9 minutes, 50 seconds here. So for those who have any questions, I'll be more than glad. And it has been my pleasure to have this small topic presentation. Hopefully that in the future we'll have more and more presentations about those things. I'm really interested more about medications. This is where my field is. So if we have anything related to even prescriptions things, over the counter, anything related to healthcare system, I'll be more than glad to present here. Thank you and have a great day.